promise that your names and your pictures will not appear in the video. If that is likely to be a problem, let me know. You can certainly, I don't have to see you to give this lecture, so you can, turn off your camera if you need to. For most, it's probably not a problem, but there's always a few where confidentiality could be a problem. So switching over to that, we have the notes right here. We've talked about eukaryotic cells and all of the complexity that they've got. That diagram shows you everything we talked about and then a few things that I think we didn't. For our purposes here, don't worry about lysosomes or peroxisomes, but ribosomes we've talked about, centrioles or centrosomes we talked about in cell division, and you can see mitochondria and the Golgi and the ER and all of that stuff. This is what we covered the last time. And this raises the question as to where it came from. And the person that worked out a big part of how this all came to be was Lynn Margulis. She died about 9 years ago. I'm proud to say I got to meet her a couple of times before she passed. She was a brilliant, quirky, but brilliant person. And in 1970, when she was still married to the astronomer Carl Sagan, who you may not remember, but he created the first Cosmos TV show, and it's something I remember growing up. But she was a great scientist in her own right, and she published a book called Origin of Eukaryotic Cells. To be fair, she built on some ideas that earlier biologists had, and really everybody builds on ideas that earlier people have had. We're all standing on the shoulders of our predecessors. But she was in a position to back up those ideas with some data from this new field that was just beginning to be called molecular biology. The study of DNA and proteins and all of these things on the molecular level. And she used those ideas to propose and support a new hypothesis. And that is that eukaryote mitochondria didn't evolve in place. They're actually descended from free-living bacteria that used to have an independent existence. They used to, swim around and do their bacterial things. Roughly 2 billion years ago, give or take a week, they entered an early eukaryotic cell, and they took up residence there. Maybe in the beginning they were parasites on the cell, maybe they weren't. There's various ways how this could have played out, but however it happened, the mitochondria developed a symbiosis with the host. Symbiosis means living together. Sym or syn is Greek for together, and bio is, of course, life. And in symbiosis, you have two or occasionally more organisms of different species living in a very tight relationship. Frequently right on top of each other or sometimes inside each other. And this particular symbiosis is a type called mutualism, a type in which both partners benefit. The mitochondria got a place to live. The earliest eukaryotes got the ability to use oxygen to break down food and generate a lot more ATP than they otherwise could. There are some eukaryotes that don't actually have mitochondria. They tend to be parasitic protists that live in odd corners of the body. But most eukaryotes use their mitochondria to take oxygen and use some chemical magic to extract lots of ATP from the molecules that they take in. So the eukaryotes gained the ability to generate lots more energy. The mitochondria got a home and got molecules that they needed. Both partners benefited. And the symbiosis has become so close that mitochondria now cannot survive outside their host cell. And most eukaryotes cannot survive without their mitochondria. They are so interdependent on each other that we think of mitochondria as being organelles, not as things that used to have an independent existence, used to be able to live on their own 2 billion or so years ago. Chloroplasts, it's the same. Those little green dots that you saw floating around in Elodia way back 1 million years ago in the second lab we ever did, they're descended from free-living bacteria that were photosynthetic, right, that took light energy and harnessed that to run their metabolism. And they also formed a symbiosis with host eukaryotes. Specifically, this type of symbiosis is called endosymbiosis. Endo means inside. So one type of cell, the ancestors of mitochondria, and also the ancestors of chloroplasts living inside another type of cell. Okay, take a few moments just to go, whoa, this is the coolest idea ever. I'm sure you all did that. Margulis's ideas were considered kind of nutty when they first came out, partly because it does seem like a lot to swallow. The idea that a eukaryotic cell isn't really one cell, it's more like a conglomerate of cells. She faced some sexism in the early days as well. This being 1970, there were still a lot of people in science whose reaction was probably something like, oh, look, it's a little lady coming up with scientific ideas. Isn't that precious? Now go make me a sandwich. I can't say it's perfect now in science in 2020, but I think it was probably worse 50 years ago. But her ideas turned out to be far and away the most widely accepted hypothesis for how eukaryotes came to be. And evidence includes the fact that mitochondria and chloroplasts have their own DNA. And when you sequence mitochondrial and chloroplast DNA, it turns out the sequences are more similar to bacterial DNA than to the host DNA in the host chromosomes. And in fact, the DNA of mitochondria and chloroplast is usually a loop. It's usually circular. Eukaryote DNA in the nucleus is strips, right? Each chromosome is a long piece of DNA with a beginning and an end. Mitochondrial DNA is like bacterial DNA in that it forms circles. It forms loops. Mitochondria and chloroplasts both have their own ribosomes. And one of the differences between eukaryotes and prokaryotes that I didn't really get into is that eukaryote ribosomes are just a little bigger. Mitochondria and chloroplasts not only have their own ribosomes, they're bacteria-sized ribosomes. They're the smaller type. And they carry out translation. Mitochondria transcribe their own genes. They make their own proteins. And mitochondria and chloroplasts can be killed by certain antibiotics that attack bacteria. Those tend to be antibiotics that are not safe for humans to use. The antibiotics that you can take to treat infections safely for various reasons tend not to go after your mitochondria or chloroplasts. But there are some that will kill bacteria, but they'll also take out your mitochondria. And if a eukaryotic cell loses its mitochondria or its chloroplast, it can't make any more. If you lost your mitochondria, you die very quickly, and your cells would not be able to manufacture any new ones. You know, once you lose them, they're gone. You get them from your mother. All of the mitochondria in your cells are descended from the mitochondria that was in your mother's egg cell. And she, of course, got all of her mitochondria from her mother's egg cell, and so on and so on, um, all the way back through the evolution of life. Dave, that's a map of mitochondrial DNA, and you certainly don't have to recognize all of the abbreviations and arrows and pointy things on it. For our purposes, it's not that important. What I wanted you to see is that mitochondrial DNA is a loop. It's like bacterial DNA. It's not like the chromosomes in a eukaryotic nucleus. Mitochondria don't have enough genes that they could survive completely independently. Mitochondria, if you took them out of a host cell, would not be able to make all the proteins they need, and they would eventually die. Maybe not that instant, but probably in a few hours. But they have enough of their own DNA that a eukaryotic cell can't make all of the proteins that mitochondria need. They still have to make some of their own. The same is true for chloroplast DNA, which is also a loop. And we can sequence mitochondrial DNA the same way we could any DNA. And you can run that through computer programs that will draw up these branching diagrams, these cladograms. And when we do this, mitochondrial DNA sequences are represented by the names in those boxes that are outlined in blue. That's DNA from the mitochondria of 3 different protist species. And it turns out that they share a common ancestor with DNA of a specific group of bacteria shown in red. Those are bacteria that are called rickettsias. If you're curious, a rickettsia causes a human disease called typhus, which is treatable now, but back before antibiotics were developed, typhus outbreaks were a real problem. Something like 25% of infected people would die of it. It's the kind of thing that you might read about in, I don't know, Charles Dickens and other Victorian novels. Oh, poor Elsie has taken sick with the typhus, and we are expecting her to perish, that sort of thing. Sorry about the crap accent. But yeah, we can sequence mitochondrial DNA, and as we talked about in an earlier lecture, trace similarities between that mitochondrial DNA and DNA of a particular group of bacteria. That's a transmission electron micrograph of a slice through a mitochondrion. It's got these internal membrane sacs where a lot of the magic that generates ATP happens. For this class, we don't really need to worry about how that goes, although there's at least a few students that were going to take this course and then move on to the major sequence. So if you're listening, you'll get the details in that course later. What you need to see for now is that mitochondria and also chloroplasts are surrounded not by a single membrane, but by a double layer of membrane. You should be able to see that thing surrounding the mitochondrion is double. And that makes sense if you assume that there was a time when mitochondria and chloroplast ancestors lived outside. They were surrounded by a single membrane, and then they penetrated into a host cell. Maybe the host tried to swallow it. Maybe they pushed their way in themselves. The details we don't fully know, but they're not critical for what we're doing here. But as they were engulfed or they penetrated into a host, they got wrapped in a second layer of membrane. That double membrane is structural evidence that mitochondria and chloroplasts used to be independent little entities, independent living things. And relationships like this aren't unusual. If we had no precedent for it, then Margulis's ideas still might not be accepted. But this kind of close interrelationship is all over the map in the living world. Way back a million years ago, when I was lecturing about evolution, I think I told you guys that I hate the phrase survival of the fittest. And the reason is that makes people think that evolution is about survival of the meanest, that only the fiercest and toughest and strongest competitors can possibly win the struggle for survival. You know, the kind of thing you get on Shark Week on Animal Planet, or is it Discovery Channel? I don't know. You know, the kind of thing you get in those nature documentaries where the shark is savagely tearing up the dolphin, or the lion is savagely tearing apart the helpless gazelle, or something like that. While I don't deny that nature can be cruel, you know, there's a reason why nature is called a mother. At least as common, and probably much more so, is symbiosis. Very, 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 very often we can see cases all around us that the way to win the evolutionary struggle for existence is to cooperate. And it's really no different in, you know, human business. Very often companies of different sorts make arrangements and agreements that benefit both of them. Sometimes companies even merge into one. And this sort of thing happens in nature all the time. On the left, you're looking at a picture taken through a microscope of little single-celled algae, little greenish-brown spots. Each one of those is just one cell. They're called symbiodinium, and they live inside the tissues of corals. On the right, you've got a living brain coral living in tropical ocean. Coral reefs won't grow if there's not enough sunlight. They have to have lots of sunlight to grow. And the reason is that the tissue of the living coral is jam-packed with single-celled algae. You can even see it's the same color. The green stuff on the right is the living tissue of the brain coral, and it's green because it's loaded with single-celled eukaryotes living inside the coral, providing food for the coral and getting a place to live and nutrients from the coral. It's a mutualistic symbiosis, and it's responsible for most of the world's coral reefs, certainly all of the tropical ones. Ding. We can find protists today that depend on symbiotic bacteria. What you're looking at is just a single-celled protist. It happens to be called Pollinella. What you need to know about it is that it can't survive without photosynthetic bacteria inside its own cells. That thing inside Pollinella that looks kind of like a green spiral, that is actually a photosynthetic bacterium living inside Pollinella and enabling Pollinella to survive. If we'd had a girl, maybe I should have named her Pollinella. It's kind of a nice name. I digress. Anyway, so things like that, eukaryotes that have symbiotic bacteria living inside them now, are common today. We can study them, and we've been able to gain some insight into what early eukaryotes were like. If nothing else, we see that it's perfectly possible for a eukaryotic cell to make a living by depending on symbiotic prokaryotes that live inside it. So Margulis's idea really isn't far-fetched, because things like this happen around us all the time. To be more specific, that type of symbiosis, again, is called mutualism. Symbiosis is any type of very close relationship between species that are not closely related. Mutualism is a type of symbiosis where both partners benefit each other. This is very common in the last really big topic of this course, ecology, which is what we're going to cover for the rest of today and for the next 4 lectures, and that will bring the course to an end. You're looking at a honeybee feeding on nectar from a flower. Many flowers make sweet juice, very rich in sugar inside, producing it from glands in the flower called nectaries, if you must know. And various insects, most famously honeybees, but also some other types of insects as well, will come along and drink the nectar. Honeybees actually depend on this because when they drink it, they'll store it, they'll come back to the hive, they'll barf it up and convert it through some chemistry into honey. And of course, humans harvest honey, but the bees depend on honey as their food, especially for surviving through the winter. So the bee benefits. The bee not only feeds on nectar, bees also feed on pollen. Pollen grains are not exactly sperm, but you can think of them as such. To reproduce, flowers have to get pollen from one flower to another. Flowers, since they can't walk around and they can't, go meet up and, try to pick up girls in bars or something like that, flowers have to have other ways of getting Mr. Sperm to Mrs. Egg. And many flowering plants use bees. This bee will eat most of the pollen, and she'll feed on the flower nectar, but she'll also pick up pollen and transfer it from one flower to another, and that's what fertilizes the egg cells of the flower and enables the plant to produce seeds and fruits. So the flowers could not reproduce without bees. The bees could not survive without the foods that the flowers provide. So that's a mutualistic relationship. Both partners benefit out of the deal. It's not only bees. Honeybees are famous for doing this, but bumblebees and carpenter bees are pollinating flowers in my yard as we speak. There's lots of other bees that don't produce significant amounts of honey, but they do pollination. Hummingbirds pollinate certain flowers, so do butterflies, moths, even houseflies will pollinate certain flowers, and bats do it as well. Not so much the bats that we get here in Arkansas, but the long-faced bats known as flying foxes that you get in the tropics feed on fruit and flowers, and they pollinate many plants. They pollinate tropical fruits like mangoes and guavas. They pollinate the orchid that gives us natural vanilla. They also pollinate agave, which means that if it wasn't for bat pollinators, we would have no tequila, and life would be so poor. There would be no margaritas if we didn't have bats. Think of that sometime in the future when you get to go to a restaurant for a pitcher of margaritas. Thank the bats. Moving right along. One more that you could probably see wherever you are, these crusty growths that are growing on tree bark are called lichens. I can see some from where I'm sitting right now. Just about any tree of significant age, certainly in Arkansas, probably in Denmark too, is going to have some of these growing on the bark, these kind of crusty, usually whitish to greenish to gray, sometimes bright orange, sometimes other colors. But just about any tree is going to have those growing on the bark. They're not plants. They're actually a mutualistic symbiosis between fungi and photosynthetic protists. The fungi get nutrients from the protists. the protists get a protected place to live inside the fungi, and the two of them together grow in ways and in places that neither one could do on its own. So when you look at a lichen, you're not looking at a single organism. You're actually looking at least two and sometimes more organisms growing together so tightly that it takes careful study to be able to figure out where one ends and the other begins. Little bit of everybody doing okay? Am I making a certain degree of sense?
Yes.
Okay. Little bit of trivia, by the way. One of the first scientists to figure out that lichens were really fungi and protists growing together and not single organisms at all was an English woman working in the 19th century. There were a couple of other people that figured it out at about the same time. I don't know who was absolutely first, but she was one of the first. And she wrote this up, but wasn't allowed to publish her findings because, being a woman, she was clearly unfitted to do science. So she'd illustrated her findings with paintings. Photography technique wasn't that great at the time, but she was a really gifted artist. So she switched over from science to writing children's books. Her name was Beatrix Potter. And I don't know if you grew up with these, but she wrote The Tale of Peter Rabbit is her most famous one, about a naughty boy rabbit who steals vegetables from Mr. McGregor's farm. She wrote one about a hedgehog named Mrs. Tiggywinkle, and one about Jemima Puddle Duck, and a bunch of others that I don't know if you know them, but I had them read to me when I was a kid. They were classics for over 100, 150 years. And while it's a good thing to write classic children's books, there's nothing wrong with that. It's kind of sad that she didn't get, the scope to publish her ideas and continue on with a career as a scientist. Says something about the way that women have not always been allowed to participate in science on an equal footing. So there's your moment of consciousness for the day. Yet another thing that fungi do, there's lots of plants. In fact, I think about 90 to 95% of all flowering plants, fungi grow into their roots. The picture you're looking at is a piece of a plant root that's been stained blue. Those boxy things that you can see, especially kind of on the upper right, are the plant cells. And then kind of scattered all through, you can see some kind of rounded blobs with little strings connecting them. That's actually fungal hyphae. And these are fungi that are growing into the plant roots, penetrating the plant cells. And fungi that do this are called mycorrhizae. Myco means a fungus, rhiza means a root. So mycorrhizae just means root fungus. And there are fungi that cause disease of plants, but these particular ones are actually beneficial. The fungi get nutrients from the plant, but the plant also gets certain minerals that it needs from the fungus. The fungus is better at picking up certain minerals out of the soil, and the fungus can pass these to the plant. And in return, the fungus gets some of the plant's sugars, some of the nutrients that the plant makes through photosynthesis. So again, this is a mutualistic symbiosis. Each partner gets what it needs. Each partner benefits. And I don't know about you, but I'm doing a lot more gardening than I used to. And it turns out that 95% of all plant species grow poorly without mycorrhizae. Even if you give them enough water and enough fertilizer, they'll do much better with mycorrhizae present. I stole this picture from the website of a company called Great White that will actually sell you mycorrhizal spores, and you can see the difference. Marigolds grown with mycorrhizae do a lot better than marigolds that are grown without. And the same would be true for most plant species. This is not an ad specifically for Great White. I'm just showing you to illustrate the difference between growth with and without mycorrhizae. Plants don't do as well if they don't have them. And again, this is not an ad, but you can buy mycorrhizal spores and add them to your potted plants, dump them into your garden, and your plants will grow much better if the mycorrhizae are thriving. I haven't done that this year. Maybe I should, but I've tried this myself. Time was you couldn't usually find these in the hardware stores. You had to go to the plant outlets on the other side of Conway to get these, but you can get them. You can buy them on Amazon, and it actually works very well, especially for potted plants that might be growing in potting mix instead of in, natural soil. So if you're doing some extra gardening these days, give this a shot. It might actually work out. See, never let it be said I don't teach useful things. There's two other types of symbiosis that we need to cover, and then that will be it for today. The second type is parasitism. And the difference between parasitism and mutualism is in mutualism, both benefit. In parasitism, one benefits and the other one is harmed. So if one is damaged by the other, then we have parasitism. These are ears of corn with these hideous gray swollen things growing out of them. Those are the fruiting bodies of a type of fungus called smut. In Latin, Ustalago maidis. In Mexican Spanish, these are called huitlacoches. And they're growing on the corn seeds. They're damaging the corn's ability to make seeds and reproduce. They'll take over all the seeds if they're not stopped. So that would be parasitism, because these are harming the corn's chances of reproducing, not giving it any sort of benefit. And, Casper, I was thinking of you because, you and your dad were going to go visit LA over spring break. I hope when all the craziness is over, you get the chance to, because one thing they've got in LA is amazing Mexican food. And huitlacoches are actually quite edible. I've had them before. They've got a very nice mushroom flavor. They're not the sort of thing that your average Mexican restaurant here is going to serve. But if you can get a place that's really authentic, they might make some dishes. Come on, Wagoner. They'll make some dishes with huitlacoches that are really good, and they're definitely worth trying. So if you ever get to a place where they get really good Mexican food, you might be able to sample huitlacoches. I think they actually sell them in Mexican grocery stores in cans. So check this out if you ever get the chance. So yeah, from our point of view, huitlacoches are beneficial. In fact, they're deliberately farmed. But from the point of view of the corn, huitlacoche is a parasite. I think the next slide is a little bit gross, so brace yourselves. Yeah, what you're looking at there is some human intestine in the middle of surgery. The person whose intestine this is has just had a whole bunch of intestinal roundworms surgically removed. This is a human parasite called Ascaris. Roundworms belong to a phylum called Nematoda, and many of these nematodes are parasites. You might know dogs get heartworm. There's other human parasites that are nematodes, like hookworm and whipworm and things like that. They take nutrition from the human host, So if you have these living in your intestine, you're not able to make use of all the food you're eating because the parasites are getting it before you can. And if you have too many of these in your intestines, they can block it completely. And if that happens, calls for emergency surgery. You're not going to live long with a blocked intestine. That's a dangerous condition to have. So certainly Ascaris harms humans, takes resources that they need, and can even potentially be lethal. So that's a nice example of a parasite right there. Yeah, sorry about that. The last possibility is called commensalism, where you have two organisms, one benefits and the other is neither really helped nor really harmed. There might be a very slight benefit or harm one way or the other. It can be hard to say that there's absolutely no benefit or harm being done ever. But if there's very little benefit or harm either way, we'd probably call this commensalism. Here you've got a cow. I assume you knew that. And the white birds surrounding it are cattle egrets. You can see these around here in Arkansas. And they'll live in cow pasture, walk behind a cow, and as the cow walks and grazes, the cow stirs up insects, and the cattle egrets will eat the insects. So the egrets benefit by hanging around next to cows. The cows don't really get any benefit, but they're not really being harmed either. So this would be a pretty good example of commensalism. The egrets benefit, the cows probably don't benefit or get harmed very much either way. To the cows, it's pretty much neutral as far as I know. All right, that would seem to be that. Got any questions before I stop recording? I can't possibly have explained that so well that nobody's got questions. I'm not that good. Okay, maybe I am that good. All right, I'm going to go ahead and stop the recording.